one of the things I'm really worried about is that the work that I do is actually useful and it's transferred to what it needs to be transferred. So I like basic research, but I also like when research has a really immediate effect, you know, and especially when we are talking about managing marine resources. So I work in ASTI, as Felix mentioned, is a technology center in the north of Spain, focused on marine and food research. And among the things we do, obviously, we are doing projects related to climate change, marine ecosystem functioning, and sustainable fisheries management. And this is what I'm going to be focusing on today. So at ASTI, we are deeply involved in the fisheries management process in several steps of the process. So this is, I know, is a very, very, very schematic view that doesn't make justice to the complexity of the fisheries management process, but it will help me walk you through this presentation, as I'm going to be giving examples on where and how in this process genetics can provide helpful information. So the first thing I want to be focusing on is on what do we assess, right? So when we do in the fisheries management process, there is of course a data collection process that will come from the fisheries and other sources, and then we do the assessment of the research that will help us providing some scientific advice that will be taken into account together with other external factors in management decisions that will, of course, affect the fisheries. So one of the questions that I think is very important and that could change everything in this process is, what do we assess? So do we assess a species, an ecosystem? And if we assess a species, do we assess a population? So what we usually use is the term stock, right? So we assess stocks in fisheries, we assess stocks. So one of the things that genetics can help us do is identify and delimiting stocks. And this, we do this using SNPs. So I'm gonna talk to you about this in particular in the following part of the talk. So stocks are basically an artificial management unit, right? So we assume that changes in stock dynamics are function on intrinsic parameters of that population, in quotes, and that such as growth, recruitment, mortality, and fishing mortality as well, and that those parameters are not dependent on emigration and immigration rates. So these management units sometimes are defined not following biological considerations. In some cases, they're just political reasons. For example, here is an example where, I don't know if you see my pointer, but here is an example where we have some fish stocks within European waters that are divided into northern and southern stock. And the existence of the southern stock is just justified because so it appeared with the annexion of Europe and of Spain and Portugal to the European Union, basically. So the obviously species and fish in particular, they don't know about boundaries at all. So is this stock separation something that is biologically meaningful? This is quite important because we actually should be managing populations. So populations and stocks should be equivalent if we want to do things right. So populations can also be defined as evolutionary significant units for management purposes. So populations would be a group of sexually interbreeding individuals which have a common gene pool. So in this case, if those two stocks, hypothetical stocks, would be behaving as a single populations, we wouldn't be doing things right because we would need to manage the population and not stocks as separate groups. So I'm going to show you some examples of case studies where we have applied genetic tools to do stock identification and stock delimitation to help the assessment of these species. So we have been working on some species that are currently assessed by ICES, so species that inhabit North European waters and in some cases, such as the mackerel, also Western Atlantic waters. We've been also working on highly migratory species, such as the bluefin tuna, which is assessed and managed by ICAT. And then we have recently also been working in the Indian Ocean with some species from IOTC. So I'm not going to talk to you about all of them. I'm just going to give you some examples so that I think I can explain how we use these techniques to improve management. So I don't want to get too into detail into the methods, but just so that you know what I will be talking about and perhaps ask later on, we'll be working with SNPs. I put this slide because I know SNP is a word that appears a lot. So just for those that are not very familiar with it. So SNPs are basically sequence variations that occur within a species. So individuals from the same species don't have identical genomes, so they have some variations. And those variations are common in individuals that reproduce within each other. And they are different in individuals that don't reproduce within each other. So we use the word genotype to specify the combination of SNPs. So we have 2 copies of the genome. So the combination of SNPs we can have in the genome. But basically, all this information that comes into the genome of a species and a population can be translated into a very, very simple table where you put the individuals and the SNPs. And then you would get the genotypes of each SNP. Obviously, this is a very simple table. Usually we have thousands of SNPs and hundreds to thousands of individuals. But this can be used to make statistical inferences. And we can use quite simple analysis as a principal component just to show how different or how similar samples are within each other, taking into account their SNPs. So here I start with the examples. So this is a work, part of a project that was funded by FAO through IOTC, also from the European Union. And that was done in collaboration with partners from CSIRO, IRD, and also Indonesian partners. So it's a project on population structure of tunas, billfishes, and sharks within the Indian Ocean. So here's the example on the big-eye tuna. So there was this concern whether the big-eye tuna, which is managed as one stock within the Indian Ocean, was composed of a single population or of several populations, and what was the relationship of the big-eye tuna in the Indian Ocean with the adjacent oceans. So we did this analysis where we collected the samples. So in this map, sorry, I went too far. In this map, you see where the samples were collected, and the biggest, the square or the circle, the biggest number of samples were collected. And the shapes indicate if the samples were adults, juvenile, or young of the year. For this type of analysis, the youngest the individuals, the better, because those are the ones that didn't have time actually to move from their birthplace. So the closest they are to the birthplace, the better to infer population connectivity. So here is the results we obtained. So as you can see, the Atlantic Ocean is very different. So samples from this area, the Atlantic Ocean, are very, very different from the samples from the Indian and Pacific Ocean. And we can also see a separation between samples from the Pacific and the Indian Ocean. And within the Indian Ocean, actually, if we do the analysis only considering the Indian Ocean, we don't see any differentiation at all. So we can actually conclude here that the big-eye tuna from the Indian Ocean is composed by a single panmitic population, we call it, and that is genetically isolated from the Pacific and Atlantic big-eye tunas. So this is perfect for stock assessment. So this is a perfect situation where we have things that makes our lives easier in that sense. So stock equals a population. So this is it's helpful. So this information will be presented to the working party on tropical tunas that will take place in a few weeks to help them focus their assessment. So in this case, their recommendation would be that they continue doing the way they are doing. Another example that is quite different is the European hake. So we come back to the Northern Atlantic. The European hake is one of those cases I was mentioning before that is separated into the northern and the southern stock, and the southern stock simply appeared just because Spain and Portugal joined the European Union. Obviously, hakes didn't know that those countries were not in Europe, so they could cross the border, I'm sure. So we had a lot of elements to think that there was a mismatch between stock and populations in this case. So this is the samples we got. So we got samples from the Norwegian Sea, Bay of Biscay, and also the Galician coast. It's not an ideal study because obviously we would have needed samples from here, but that was a small project we had, and it was a way of starting to hint how the situation was in this case. So what we saw is actually that samples from Norway formed a genetically isolated population from the samples, including the Bay of Biscay and Galician coast, and obviously they all were different from the Med. So what is interesting here is that we have for management purposes, the Bay of Biscay samples and the Norwegian samples are part of the same stock, but they are very different. And also for management purposes, samples from the Bay of Biscay and Galicia are part of two different stocks, but they are actually the same population. So this is a case where things are not right. So the stock delimitation should be somewhere here in between. We don't know where. We are doing additional analysis with additional samples to elucidate where this boundary should be. But actually, it shouldn't be where it is right now. So basically, what we have is that the southern part of the northern stock and the southern stock are part of the same population, which is genetically different from the northern part of the northern stock. So we have a case where stocks are not equal populations. So we are working together. The stock coordinator of the northern hake is part of our institute, and also we work closely with the southern stock coordinator. So we are in contact with them, and they are aware of this situation, and we are working on trying to see how the assessment would change if we change those boundaries. And in parallel, we are also doing additional genetic analysis to further understand where the stock boundary should be. Okay, another case, Atlantic bluefin tuna. This is a very, very exciting species, I would say, because both from management, conservation, biology, is a very emblematic species. So it's a very nice case study. So the bluefin tuna is assessed by ICAT as two stocks that are divided by the -45th meridian. So whatever is caught in the east is considered eastern stock, and whatever is caught in the west is considered western stock. This is based on the assumption that the bluefin tuna, and the current knowledge that the bluefin tuna spawns in the Mediterranean and in the Gulf of Mexico, although there is a new spawning ground that was recently discovered in the Slope Sea. So we assume that the individuals caught at each side of this line are born in the spawning grounds that are at each side of that line. But obviously we know that the bluefin tuna don't care about this line, and they can cross the Atlantic even several times in their lifetime. So we know that we can capture bluefin tuna in Labrador that was born in the Mediterranean. So what do we do about this, right? So we got this project that was funded by ICAT in order to determine what was going on in the bluefin tuna, and if we could provide some tools to facilitate the assessment of this species. So we collected young of the year and larvae from the spawning grounds, because those are the ones that actually didn't move or didn't have time to move from where they were born. And we performed some genetic analysis. And as you can see here, the separation is not as clear as in the hake, but you can see that there is a separation between the individuals born in the Mediterranean and the individuals born in the Western Atlantic. So we see that there is a separation. So there seems to be some reasoning for having two stocks. However, we don't know if the way we are assigning individuals to stocks is correct. So yes, there is differentiation between Western and Eastern spawning components. And yes, there should be two stocks, but not separated by a line. So in this case, again, the stock doesn't mean population. So what we actually did is that we developed a tool based on this genetic differentiation. we can develop a tool where we analyze a few of those SNPs in each individual, and we can assign where they were born. And this is what we did. And this is the, so in these pie charts, what you can see in orange is the proportion of individuals that genetically were Mediterranean-like, and in purple, the proportion of individuals that were genetically Gulf of Mexico-like. In gray, there are individuals that we cannot assign to either spawning ground. We have some explanations for this because this is work ongoing. I don't have time to present that other part, but we are discovering very exciting things on this species, and it seems that the situation is not as simple as we put it in this slide. But for management purposes, this is very helpful because it actually shows them that this is actually something that was already thought before, that the proportion of Mediterranean individuals in Western waters is quite high. So this should be taken into account in the assessment. And this information is actually taken by ICAT, and they are actually now including it in the assessment and the MSc and so on. So it is a case where the genetic information is actually providing useful advice on how to do the assessment. So in our ASTI communication team, they call this the genetic birth certificate of Atlantic bluefin tuna, and I thought it was very cool. Okay, so in this case, with this tool, yes, we can say that in this case, stock equals population because we can assign individuals to their stock. So we don't assign individuals to stock now based on where they were captured, but on where the genetic tells us they were born. Okay, and this is the last example I will be focusing on. It's the white anglerfish. I'm going to be talking a little bit longer about this species because we found something we were not looking for. And this is an example of where genetics can not only sometimes make you have information in an easier, perhaps a cheaper way, but it can also show you things that you cannot see otherwise. So in the case of white anglerfish, it was a project funded by the JRC from the European Commission. And actually the first motivation for working with this species is that we wanted to apply the close-kin-mar recapture to this species. So this is a method to determine spawning stock biomass independent of the fishery. And I will talk to you a little bit later about this. But in order to apply the close-kin-mar recapture method, you need to actually know if your the area you are covering with the study is connected or not. So the same thing, you need to assess if stocks equal populations. So we sampled individuals throughout the distribution of the white anglerfish. So the white anglerfish is assessed as 3 stocks. So the northern platform stock, the northern stock, and the southern stock, and then we have the Mediterranean. We also sampled individuals of black anglerfish. They are sister species, and we sampled just a few of them to have them as an outgroup. The two species are very similar, but they are identified in base of the color of their peritoneum. You can see here is this epithelium here that is black in the black anglerfish and white in the white anglerfish. So here is the result we got using those nuclear SNPs. So it's a PCA that doesn't look at all as the other PCAs I showed you before. This is you can see that there are a lot of dots just here, a lot of dots just here, some of them here, and then some separated ones here. So it seems a bit random. The first thing we did was to see if there were some problems, you know, in the lab and everything. You check everything when you get a strange result like this. But it turns out that this is something that has a biological explanation. So in this mountain of dots, stack of dots, we have individuals from the 3 stocks and also from the Mediterranean. In this middle kind of a stack of dots, we also have individuals from all the stocks except the southern and the Mediterranean. And in this other pile of dots, we have also individuals from all the stocks except from the northern platform and the black anglerfish individuals. So, and then, yeah, sorry, I forgot about those. And those ones were part of the northern stock. So this was a bit confusing to us. But the fact that the some white anglerfish grouped exactly where the black anglerfish grouped was a little bit suspicious of a potential misidentification problem. So we actually used a mitochondrial marker that has been published that can identify which individuals are white and which individuals are black anglerfish. So we found that all the black anglerfish were actually correctly typed in using the mitochondrial DNA, but the white anglerfish, some of them seem to be white and some of them seem to be black. So what we have here, so we have a completely different problem, and it's that we have a problem of a species identification. So not only a stock is not a population, but it's not one species. So we have more than one species that are assessed together without us knowing it, which is the main problem. So basically, in summary, what we have is that what it is landed or what is caught as a white anglerfish could actually be either a black anglerfish, misidentified, so a black anglerfish with a white peritoneum, could be something that perhaps there are hybrids, or could be white anglerfish. And whatever is caught as black anglerfish is black anglerfish. So black is always black so far, because we haven't sampled so many black anglerfish. So we cannot discard that the problem is the other way around. So basically, those individuals would be real white anglerfish, Lophius piscatorius. Those individuals would be black anglerfish, misidentified, Lophius budegassa. And then the ones in the middle are probably hybrids. So there is some test we can do, and actually it turns that they are hybrids. So we can have hybrids between white and black anglerfish. This was not known. This was something that we discovered just by doing this analysis that were actually planned for something else. And also what we find also is that we have some back crosses. So those individuals here in between are actually back crosses. So a hybrid with a pure. Those are called back crosses. We don't find anything else. We don't find any individuals that would go below here. So we don't have. We only have hybrids, half, and then the back crosses. So in this map, you see the distribution of the mislabeled and hybrid individuals, colored by stock for the 3 stocks. So the mislabeled individuals are in black. So those are individuals that are given to us as white anglerfish, but they are actually black. You can see the proportion in the south is quite high, and in the Mediterranean is very high, considering the low number of individuals that have been sampled. And also in the Bay of Biscay and Celtic Sea, the number of hybrids is the highest. So for management purposes, well, this is a problem because we have a percentage of the individuals that we are considering white anglerfish that are not white anglerfish. So we don't only have a problem with the species identification, we also have the species of hybrids that are reported as white anglerfish. What is interesting is that the hybrids occur in this area. And as you can see in these maps, this area here is where the 2 species co-occur for a long time. So this is the evolution of the distribution of the white and black anglerfish through the years. So from 1995 to last year. And you can see, well, as many species we know, they are moving northwards. Both species are moving north, but since a long time ago, the 2 species co-occur in the area where most of the hybrids occur. But we know that this is a changing situation. So one could anticipate that perhaps hybrids that are not currently found in the North Sea, for example, will be found in the future. So speaking about future, one of the things is what happens with hybrids, right? What is the effect they will have in those species? Will they have an effect? Should we worry about them? So one thing is that we can have is that, well, currently we know that the hybrids are occurring, that's for sure. We don't know if they occurred in the past, and we don't know if they will occur in the future. So if they are old, if they have been occurring for a long time, well, we could think about a stable hybrid zone. So the 2 species exist. There is a hybrid zone. Hybrids are less fit. They cannot really reproduce except for some exceptions. So they are not really, you know, continuing. So they will stay there as they are. And if they are recent, well, they could be also that we are in the situation where we have a stable hybrid zone. Hybrids will coexist with the other two species. Or we could have a situation where the hybrids could create a new species. So the species, black and white as we know them now, will disappear. They will start mixing with each other, and then a new species will appear. Oh, sorry, one species. Or that the hybrids will form actually a third species that it's different from the other two. We don't know. We don't have now the tools or the information to know what will happen in the future. But for sure, we need to keep monitoring these hybrids to see if they are increasing or they are stabilizing or what's going on with them. Once we clean everything and we keep only the individuals that are actually white angler fish, we do a population genetic study as we have done in other cases, and we see that the Atlantic is separated from the Mediterranean and that the Atlantic forms a single panmetic population. So there is no reason for having 3 stocks in the white anglerfish, even if we remove all the problems with identification and hybrids. So in this case, again, we have a problem of stock doesn't equal population. So this is also, again, something that we are working with the assessment groups in ICES, and they are aware of this information. We have presented this information to them, and we are working on trying to see how we can include this information in the assessment, or how we can inform the assessment at least of this, this problem here. So this was about the stock delimitation identification of SNP. So this was the longest part of the talk. So I'm going to briefly talk to you about 3 other things. One is, I mentioned, well, the data collection relies on fisheries, but obviously it can also make use of fishery independent data. We have surveys and we have many different data we can use. One of the things that is very now people talk a lot about this method in terms of improving fisheries management is the CKMR recapture. It's spawning stock biomass estimation that is independent from the fishery, and that is based on determining kin relationships between individuals. So this is a very cartoon example of the method, but helps understanding it. So we can imagine here 2 populations of fish. Obviously, we cannot see them, so we don't know how big they are or how small they are. So if we sample randomly adults and juveniles, we would find pairs of related individuals. In the small population, the number of pairs of related individuals, parents and children, we will find will be larger than in the large population. So by knowing how many pairs of related individuals we find with a given number of individuals sampled, we can make an inference on how big or how small the population is. You can imagine a small village or a very big town, right? If you be a very big city, if you sample 10 individuals of each, the probability of finding relatives in the small town will be higher. And then obviously there are some formulas. This is a very, very simple, and there are many parameters that need to be taken into account that are specific for each species. But basically the principle is this one. So in order to apply closed-KIN-MAR recapture to a species, there are several requirements that need to be fulfilled. So here is the reflection we did for the white anglerfish. So, well, the things that you need to consider before, so is the species breeding biology appropriate? Obviously, those species that are very short-lived or those species that breed and die and so on, you cannot use them for closed-kin, but this is not the case of the anglerfish. Is there enough biological knowledge available? Well, we need to know things in order to apply the closed-kin. We need to know things such as maturity at age. We need to know, how large would be the offspring. We need to know the longevity, a lot of things. In the anglerfish, well, we could know more, but we know some things. Sampling and logistics are appropriate. Yes, sampling is currently done. Genetic population structure, no, I just presented that. And then is there a realistic estimation estimated number of samples needed. So how many samples do we need to apply this method? And this is something we can actually calculate. And this is something we have done for anglerfish. So you can do scoping study. You can enter a lot of the parameters you know from that species. We have done this for the northern and the southern stock separately. And then you can estimate how many samples you would need in order to obtain a given number of related pairs. So here is the number of samples you would need and the number of related pairs. You can see that for the southern stock, we need to sample much lower number of samples to obtain 50 pairs. And for the northern stock, we need to sample more. But all these samples are in the range of the samples that are actually being collected for estimating lengths and so on. So this is something that tells us that we could actually apply this method. And this is basically the same thing, but playing a little bit with the proportion of adults and juveniles with samples. So you can really play a lot with the data. But the basic message here is that for white anglerfish, there could be the number of samples required is actually something that is logistically possible as things are currently. And after that, obviously, we need to develop the statistical model and so on, but we haven't started that yet, so we are currently on that part. Okay, so another thing. Yes.
Sorry, science is great, but can you wrap it up?
I will do it. This is very fast. Don't worry. This is very, very fast. So another thing we can do is, well, the assessment. Now I've been talking about assessing one single species, but obviously now we are moving towards ecosystem-based assessment. So we need to assess more things. And among the things we need to assess is we need to consider food webs. So using genetics, we can analyze stomach contents and we can know which species are in the stomachs. I stop here. Yeah, so this is interesting, obviously, for estimating natural mortality, for multi-specific models, and so on. There are some drawbacks. So using genetics, we cannot detect cannibalism, we cannot distinguish, larvae, juveniles, so on. But the method is cheap, doesn't require taxonomic expertise, performs accurate ID in digested prey, and performs accurate ID in in larvae or eggs, and so on. Another thing, last one, and this is very short at the end, is on eDNA. Here it comes. So yeah, so this is related obviously with the diversity and biomass estimation, so with the fisheries independent data, but it's also related to the ecosystem-based fisheries management because you take into account the whole ecosystem and all the species. So eDNA is basically those traces of DNA that are occur in the environment and they're released by microorganisms. You can collect eDNA from water by filtering water. You can sequence this eDNA, can compare it with a reference database, and you can obtain an inventory of the species that live in that environment. So this is, well, I'm not going to go through this, but I leave it here just in case we have questions. So there is a lot of things that we need to consider when we study eDNA. And this is something we call the ecology of eDNA. So DNA doesn't move the same way as the species from where it comes from. And this needs to be considered when we do the interpretations. So this is a study we did in the Bay of Biscay where we compare eDNA versus trawling. So here is the results. So eDNA, basically more than 100 species, trawling very few, and the most common ones are in common. The ones that we don't catch with eDNA, some of them are not in reference databases, they are very low abundant, and so on. There is a relationship between the number of copies of eDNA and the biomass of those common species. So this is quite interesting. And also there is a correlation or relation between how far the sample was collected and the number of copies of eDNA, and this is congruent with the lifestyle of those species. More coastal species are found in more shallow stations, and more deep species are found in locations that are farther away from the coast. We are working now, and that's why I put this little image here, on using eDNA to study deep sea diversity, in particular mesopelagic fish. And obviously I don't have time to present that, so I finish here with, I would like to emphasize that obviously this is teamwork. I'm the one presenting it, but I put here so the pictures are from the people that were leading at least one of these studies, but obviously they were not doing it alone. So we have a lot of input and help, and we work a lot with people from the marine ecosystem functioning and sustainable fisheries management area, and with external collaboration, which I don't have time or space to name them all. So this is the end. Thank you very much for listening.